Hello everyone, very warm welcome to all of you. This is part 5, chapter 8, unit 3. In this part, we will discuss about ribosomes, 70S ribosome and 80S ribosomes, different type of cytoskeleton found in eukaryotic cells. Then we have to see what we mean by cilia and flagella, their function, structure and differences. And in the last, we will see centrosome, centriole, structure and their function. First, we have to discuss about ribosomes, also known as pallet particles, as were discovered by Pallet. Ribosomes are granular structure observed first time under electron microscope by Pallet, therefore known as pallet particle. These are granules made up of only two things, that is ribonucleic acid, which we used to write that is ribo, that is nucleic acid, and ribosomal, right, and protein. They do not have any membrane. These are membraneless particles. Two important question. In case of eukaryotic cell, the ribosomes are of 80S there, that is 𝕍𝕀𝔻𝔼𝕆 unit this is, while in case of prokaryotic cell, the ribosome are 70S. In prokaryotic, this is 70S, while in case of eukaryotic, this one is 80S. You see, each is made up of two subunits. 70S is also made up of two units, and the same way 80S is also made of two subunits. The two subunits of 70S, these are 30S and that is 50S, and in case of 80S, the subunits are 40S and 60S. Remember, don't get confused. This is not sum, that is 30 plus 50 that should be equal to 80. No, it is different. That is their 𝕍𝕀𝔻𝔼 unit, sedimentation coefficient. This is, you see, the two subunits 80S I told you made up of 60S and 40S, while 70S ribosome is made up of 50S and 30S. Definitely this is smaller unit, this is bigger, this is smaller, and this one is bigger. In case of eukaryotic cell, 70S ribosome are also found. Very important point here. You see the important question. What is the important question? Ribosomes is the only organelle found in both, that is prokaryotic cell as well as in case of eukaryotic cell. Ribosome is found in both. No doubt, in case of prokaryotic cell, this is 70S, while in case of eukaryotic cell, this is 80S. But one more important question, the eukaryotic cell, it has 80S as well as 70S ribosome. Very important point, what we mean by, if this is a eukaryotic cell and here in cytoplasm of eukaryotic cell, this one is 80S ribosome, while in case of that is, let this is mitochondria, or let this one is that is chloroplast. In mitochondria and chloroplast of eukaryotic cell, they also have ribosome in it, and this ribosome is 70S type. Important question, I repeat, eukaryotic cell 70S ribosome are found in chloroplast and mitochondria, and 80S is found in cytoplasm. Therefore, these organelles are known as ribosome, are called organelle within organelle. You know, mitochondria and chloroplast, these are organelle. Inside organelle, the other organelle is found, that is 70S ribosome. Is it? So ribosome, I told you, composed of two units, larger subunit, it is dome shaped, while the smaller subunit, that is, it occurs in the form of cap-like structure. These are negatively charged molecules and, you know, made up of ribosomal RNA, and that is protein. Both subunits, they remain united with each other. Specific concentration of magnesium ion should be there. If concentration decreases, the two units will dissociate. Ribosomal subunits are separated when the concentration of magnesium ion reduces below a critical level. During protein synthesis, like this is messenger RNA, you see that is on messenger RNA, many of the ribosome, they will be lined up like this, and such a structure is known as polysome or polyribosome, or also called as that is ergosome. If we see detailed structure of ribosome, In case of prokaryotic one, that is, it is having 60:40, 60 that is ribosomal RNA, 40 that is protein. Ribosome is made up of that is 30S and 50S unit, smaller unit and larger unit. It consists of, smaller unit consists of 21 type of protein and it contains 16S ribosomal RNA. Very important one. You have to say that is this RNA, it plays important role during protein synthesis. In the same way, the larger unit of prokaryotic ribosome that is consists of 34 protein and 23S and 5S that is ribosomal RNA. It means in prokaryotic cell we have that is three type of RNAs. One is that is 5S, one is that is 16S, one is that is 23S. These two are found in larger unit while this one is found in that is smaller unit. Is it right? Yes. Now you see ribosome, this is structure of ATS ribosome. The same thing, little different. 40:60 ratio is of that is RNA and protein made up of two units. 40S is the smaller one, 60S is the larger one. This consists of 33 protein molecule and that is only one type of ribosomal RNA, that is 18S ribosomal RNA. In the same way it consists of 40 types of protein and 3 type of RNA, that is 5S, 5.8S and 28S ribosomal RNA. In case of prokaryote, ribosomal RNA and protein are synthesized in cytoplasm, but in case of eukaryotic, ribosomal subunits are synthesized in nucleolus. Here in case of eukaryotic cell, you know, this is nucleus. Inside nucleus, this one is nucleolus. That is ribosomal RNA will be synthesized in nucleolus and then they will be transferred to cytoplasm where this ribosome will be formed. While in case of prokaryotic cell, because we do not have nuclear membrane, therefore this ribosomal RNA and protein synthesis both will take place therein, that is cytoplasm. Functions of ribosome, that is everyone knows, that is this is the site of protein synthesis, known as that is this is factory for protein synthesis in cell. You can see the diagram here. This is larger unit, this smaller unit, this is the complete that is ribosome. You see the summarized account. Prokaryotic one, smaller unit, larger unit consists of 16S, then ribosomal RNA, 21 type of protein. Larger unit consists of 23S and 5S ribosomal RNA and 34 types of protein. While in case of eukaryotic one, smaller unit consists of 18S ribosomal RNA and 30 types of protein. In the same way, larger unit consists of 3 RNAs, 28S, 5.8S, 5S, and it consists of 40 types of protein. Is it okay? Come to the next now, we have to see cytoskeleton. Cytoskeleton, you see, one very important point. As the name indicates, the skeleton which will provide rigidity to the cytoplasm, as skeleton provide that is support to our body. In the same way, cytoplasm is supported by these structures known as cytoskeleton. You know, cytoskeleton is found only in a eukaryotic cell. Remember, only eukaryotic cell has that is cytoskeleton present in cytoplasm of eukaryotic cell. It is made up of extremely minute fibrous filamentous tubular proteinous structure, and that will make the structural framework inside the cell, which will provide different type of support and also perform different type of functions. You can see that this is the eukaryotic cell and this is the cytoplasm of this. So here we have 3 type of cytoskeleton. One is called microtubules. You know, that is this, in some cases that is cilia are present or that flagella are present. That is these cilia and flagella, that is they occur in cilia and flagella, in centriole and in basal body. You know, cilia and flagella, from where they arise, these are known as basal bodies, and that is, you know, the centriole is there, that is close to the nucleus. These are the centriole. So all these that is cilia, flagella, centriole, basal bodies, that is they are made up of microtubules, and even micro, that is mitotic apparatus. You know, at the time of cell division, that is the chromosomes will be arranged there at the equator line, and they will be, that is, move towards the pole. That is, these are the spindle rays, and spindle rays are also made up of microtubules. So these are hollow, unbranched cylindrical structure. The diameter is of about 25 nanometer, and with the hollow core is about, that is 25 nanometer. This is the microtubule. This is that is cavity we have here. Cavity is made up of about 50 nanometer, while the diameter of this is about 25 nanometer. So this microtubule is hollow structure. Is it clear? Come to the next now. We have to see microfilament. These are solid. Remember, microtubules were hollow. If I draw here, that is, this was the microtubule, hollow structure it was, while this microfilament, these are not hollow, these are, that is, solid structure like this, solid rod-like structure of indefinite growth. The diameter is about 6 to, that is 8 nanometer is the diameter from this to this. They are mainly composed of globular protein, that is actin, and that is filamentous myosin protein. Remember, myosin and actin, these are going to make this microfilament. They provide support to plasma membrane. These are involved in cytoplasmic streaming movement, and they help in pseudopodia formation. See, one by one, support to plasma membrane, that this is plasma membrane of a cell, so it will be supported here by, that is, this microfilaments. Cytoplasmic movement, you know, that is around the vacuole, cytoplasm used to move in either in clockwise direction or in anti-clockwise direction. That is supported by microfilament. And in case of pseudopodia formation, for example, amoeba, a eukaryotic cell, when it wants to move in a particular direction, then what happens? Pseudopodia is to be formed. In formation of pseudopodia, these actin and myosin filament, that is microfilaments, they help in formation of pseudopodia. Are you getting my point? Come to the third type of cytoskeleton, known as intermediate filament. These are hollow, non-contractile one, made up of acidic protein. The diameter is about 8 to, that is 10 nanometer. Children, you have to remember that this cytoskeleton detail is not given in NCERT, but important, that is what is the structure and function. That's why brief account is very much essential, and I'm going to discuss. These are made up of neurofibrils, found in neurofibrils, that is axon and dendrons. You know that nerve cells is like this. This is the dendron like this. These are the axon. These are made up of intermediate filament and nuclear lamina. Nuclear lamina means, that is, this is the nuclear membrane. Below this nuclear membrane, we have a supporting structure known as, that is, nuclear lamina. It is also made up of, that is, this intermediate filament. They are involved in formation of scaffold of chromatin and forming a basket around the nucleus. Is it right? So this is about our cytoskeleton. Come to the next now. We have to say, that is cilia and flagella, structure of cilia and flagella. You see, these are outgrowth of the cell membrane. You know, that this is a cell. I'm going to take, this is a euglena we have here, like this. This is euglena. This is the plasma membrane. From plasma membrane, the extension will be there. This is known as, that is, flagellum. While in case of, this is paramecium. In case of paramecium, that is so many minute, that is, outgrowths will be there, which are coming out from, that is, plasma membrane, that is cilia. Both of these, cilia and flagella, structurally these are similar, and they cause movement of either of the cell or, that is, in surrounding fluid. These are the locomotory organ. Cilia and flagella both are, these are responsible for, that is, these are locomotory, that is, organs these are. So you see, this is the case of euglena. This is the case of paramecium. Can you differentiate, children, now? That is, what is the difference in flagellum and in cilia? Can you tell me right now? That is, this is flagellum and this one is cilia. This is found in euglena and it is found in paramecium. This one is flagellum. Singular will be flagellum, plural of this will be flagella. So you see the table here. Flagella, better to call flagellum, and cilia, that is cilium. That is, what's the difference? Flagellum number is comparatively less. In euglena you are seeing only 2 are there, 1 is visible. Number of the flagella per cell is usually 1 to 4, while number of the cilia per cell, it is very large. It may be even hundreds, even 4, 5 and even more, 4, 500. They are longer in size, you are seeing. These are smaller in size difference is there. Flagella are commonly found at one end of the cell. You know that this is anterior end of the cell. This is found usually at one end, while cilia, these are found throughout, that is, or major part of the surface of the cell. They help in locomotion. They also help in locomotion, feeding, because when they will move in a synchronized way, the cilia will generate current, and this, with the help of this current, the waterways, they will be coming towards this, and that is food particles will be taken in. That is, cilia help in locomotion, in feeding, as well as that is circulation of this water towards this organism. Okay. Come to the next now. If you see the structure of cilia and flagella, you see now that is structure. Only the electron microscope has revealed structure of cilium or flagellum. As I told you, cilium and flagellum, the structure is same. There is no difference. Structure is same. So that these are covered with plasma membrane. Definitely, you see, if this is the plasma membrane we have here, then this plasma membrane extension will be there, and in that way the cilium will be here inside. Here will be the basal body, and that will be cilia or flagella. It means cilia or flagella, this is covered by plasma membrane. You see, if we cut a transverse section of this like this, that this is the flagella here. If you have cut the transverse section, then it will be like this. This is the plasma membrane outside, and you see that this is having what? How many, that is, tubules we have at the periphery. You see, this is the peripheral microtubules. Doublet is there. This is 1, this is 2, this is 3, this one is 4, this one is 5, this one is 6, this is 7, this one is 8, and this one is 9. How can we remember that there are, that is, 9 peripheral microtubules, and each is made up of 2, that is doublet. And these are connected by, that is, interdoublet bridges are there. At the center also we have central microtubules, which is made up of, that is, 2, that is, units. This is central sheath. This central microtubule is surrounded by central sheath, and this is a radial scope, that is, these are connected with the central sheath. Is it right? So such a structure, you see, the core of this, you see, I can recall you here that this is the flagella or cilia. This is the core of this. Core is known as, that is, axoneme. Core is known as axoneme, right? A number of microtubules running parallel, that this is 1, like that this is the 2. These are the microtubules running parallel to each other along to the long axis. These are lying parallel to the long axis. Axoneme usually has 9 doublets of radially arranged peripheral microtubules. As I told you, this is 1, this is 2, that will be, that is the third one, that will be the fourth one. All these are lying, that is, parallel to the long axis. Such an arrangement of that is nemal microtubule, is referred as 9+2 arrangement. Very, very important one. Here we have 2 we have at the center, and 9 we have at the periphery, starting from 1, 2, 3, 4, 5, 6, 7, 8, and that one will be 9. So such arrangement is known as 9+2 arrangement. Remember, I can recall that this is plasma membrane, and we have 9+2 arrangement. The central tubules are connected by bridge and is also enclosed by central sheath, which is connected to one of the tubule of each peripheral, that is radial. That is, this is that is doublet, and that is peripheral one. This is the central one. This is that is radial, that is radial spoke is there. This is the central sheath, right? No need to go in much more detail. Only that will solve the purpose. Both cilium and flagellum, they rise from centriole, and that is structure now known as that is basal bodies. You have to remember, in prokaryotes, that is in bacteria also, that is flagella are there, but these flagella are made up of, that is, these are different in structure, and these are made of, that is, flagellin protein. Flagellin protein in case of, that is, prokaryotic cell, while here these microtubules, these are made of, that is, tubulin protein. This is tubulin protein, okay? Different from eukaryotic flagella. Is it? Come to the next now, we have to say that is centrosome and centriole. Centrosome was discovered by Bandon and named by Boveri. Centrosome is an organelle usually containing two cylindrical structure called centriole. I may recall you here that this is the animal cell we have here, right? That this is the nucleus we have. Close to the nucleus, there is that an area is there. This area is known as centrosome. Is it? In this centrosome, we say that there is lying two cylindrical structure. One structure is this one cylindrical structure, and other cylindrical structure, it lies parallel to this, and these two cylindrical structures are known as centriole, right? You can see here, this is, that is, one centriole, this one centriole, and this is that is other centriole, and this whole area, that is known as, that is, centrosome. Is it? These centrioles, they lies at about 90 degrees to each other. If you want to say that is detailed structure of this one centriole, you are seeing that this is that is transverse section of the centriole here. If you want to enlarge this centriole, that is this central structure or transverse section of this, to see the internal structure, then such type of structure we will be able to see. You see here also that this is the periphery. On periphery we have, that is, these are the tubules. This is tubule number 1, number 2, number 3, peripheral. These are lying at the periphery of this. Are you getting my point? You can just to facilitate that, let this the structure like this. This is a hollow tubular structure. 1, 2, 3. This is 4, this is 5, this is 6, this is 7, this one is 8, and this one is 9. There are 9 structures at the periphery, and each that is made up of 3 subunits. These subunits are A, B, and C. The C of 1, that is connected to A of the conjugative, that is tubule. At center we have a central axis, but we do not have any microtubule. Therefore the structure of that, how much that will be, what we can call to this structure, we can call that arrangement is 9 plus 0 arrangement. Do you remember in case of cilia, the arrangement was, that is 9 plus 2, and here the peripheral, that is microtubules, was having 2, that is subunits. Here it is having 3 subunits, A, B, and C. This is TS of, that is centriole. Is it clear? Come to this, that is, these are surrounded by pericentrial material, centriolar material. Both the centriole in a centrosome, they lie perpendicular to each other, as I told you, which each has an organization of cartwheel type of structure. Important point, cartwheel, that is related to centriole. Cartwheel type of structure is found in centriole, made up of 9 evenly spaced peripheral fibrils made up of tubulin protein, as I told you. Each of the peripheral fibril is triplet, that is, you must be remembering one, that is, this is one. What is this? This is peripheral fibril, and this is the other peripheral fibril, that is having 3 units. This is A, B, and C. That this is A, B, and C. That this is 1 and this is 2. That is A is connected to C like this. Is it? So this is the central part of the proximal region. Centriole is also proteinase called hub. which is connected with the tubules, peripheral tubules, by radial spokes that is made up of protein. The centriole form basal bodies of cilia, as I told you there also, that this is flagellum. Here it arises from the centriole, and the centriole here it is known as, that is basal body. Okay, and spindle fibers that give rise to spindles apparatus, that is cell division. Here in case of animal cell, you know, centriole cell is found only in animal cell. It is found only in animal cell. Some of the lower plant cell which are motile, they also have centriole, but that in a typical plant cell, centriole is absent. Okay, spindle centriole form basal body, that is spindle fibers, that from where the spindle apparatus during cell division, which are responsible for chromosomal movement. You must be remembering that this is the centriole. Centriole will divide the time of division, will reach to the opposite pole. From here these spindle fibers will arise, which will draw the chromosomes towards their pole, spindle fibers. Is it okay? This is part 5. Thank you.